So the last stage of respiration is the electron transport system, and the electron transport system, it occurs in the inner membrane of the mitochondria. So the inner membrane, the mitochondria has two membranes. So the first membrane is this one, which is the outer membrane, and the inner membrane, it is this. So the inner membrane is highly folded. It is highly folded to form what you call the cristae. So this cristae here, it is where the electron transport system occurs. So one of the adaptations of the mitochondria is having the cristae, which is highly folded, to provide a large surface area for electron transport system to take place, or cellular respiration take place. Of course, cellular respiration begins from the cytoplasm. So when you look at our mitochondria, this mitochondria outside it, which is going to be the cytoplasm, because this mitochondria is found inside a cell. So outside, we are going to be having glycolysis. So outside, what is surrounding the mitochondria, it is the cytoplasm. So glycolysis is occurring in the cytoplasm. Then as we enter inside, so the inner mitochondria, so the inner mitochondrial membrane, or this membrane here, the inner membrane, that is where we're going to be having our electron transport system, and our Krebs cycle is going to be occurring in our mitochondrial matrix. So the mitochondrial matrix is located inside. So it is located inside the mitochondria. It is like the cytoplasm of the mitochondria. So it is where the Krebs cycle is going to be taking place. So in between the two membranes, we are having what you call the intermembrane space. We are having what you call the intermembrane space, and we are going to see what is going to be taking place generally from there. So if you look at the electron transport system, it marks the final stage of cellular respiration. Cellular respiration has mainly three stages. That is glycolysis, which takes place in the cytoplasm, then Krebs cycle, which takes place in the inner mitochondrial matrix, or in the mitochondrial matrix, and then lastly is the electron transport chain, which takes place in the inner mitochondrial membrane. So the inner mitochondrial membrane consists of very many foldings called what? The cristae. So it occurs in the inner mitochondrial membrane, which provides a large surface area for ATP production. So the foldings that are found in the inner mitochondrial membrane provide a large surface area, for ATP production. So we can look at the purpose of the ETC. So the ETC, or the electron transport chain, transfers electrons from NAD. So this NAD is the reduced one, and the reduced FAD to oxygen. So where are we getting the NAD from? So NAD, the reduced NAD, which is having hydrogen, is coming from glycolysis. You can also have it from the pyruvate decarboxylation in the link reaction. So we also have formation of NAD, and then you also get it from the Krebs cycle. So we are getting normally NAD in three stages. So in the glycolysis, so we are looking at reduced NAD from the glycolysis, from the link reaction. You remember the decarboxylation of the pyruvate, so we obtained NAD, and then even from the Krebs cycle. So that NAD is going to be used as a source of electrons. So we know very well that addition of electrons, that is reduction. So this is reduced NAD. And then reduced FAD, we don't find it in glycolysis, neither we do find it in the link reaction. We only find it in the Krebs cycle. So in the Krebs cycle, that is where we're going to find the reduced FAD. So reduced FAD is containing electrons. It is electron rich. So reduced NAD and reduced FAD, they are major source of electrons that are going to be used in the electron transport chain. So Purpose of the ETC. So we have looked at the first purpose, that is the transferring of electrons from the NAD, reduced NAD and reduced FAD. What are the sources of electrons? The sources of electrons, that is reduced NAD and reduced FAD. So point number 2, uses released energy to pump electrons. We are going to see how is that energy created to pump protons, not electrons, to pump protons. So we can look at number 3, creates a proton gradient used to synthesize ATP. The proton gradient that we are going to see, we know very well that protons are positively charged. So when we have a bigger positive charge created on one side and a less positive charge created on another side, we are going to have a potential difference created, of which that one is going to generate electrical current, of which electrical current produces energy, which is going to be used to synthesize ATP. So what are the major components of the ETC? The major components of the electron transport chain. You can look at the electron carrier complexes. Electron carrier complexes, we're having about 4 complexes, that is complex 1, complex 2, complex 3, and complex 4. So those are the major complexes, electron carrier complexes, that are found in the ETC. All these complexes, we find them in the inner mitochondrial membrane. So in the inner mitochondrial membrane, we are going to see that inner mitochondrial membrane. So if you look at complex 1, complex 1 is called NAD, so reduced NAD dehydrogenase, or you can also call it in some sources they can call it reduced NAD oxidoreductase. Oxidoreductase, it is just an enzyme found in complex 1, but now that complex again contains Flavin Mononucleotide, and then iron and then sulfur proteins. We shall find them there. So Flavin Mononucleotide, these are just going to be receiving electrons and then carrying them to the next component. So FES, iron sulfur containing proteins. So still these ones are going to be carrying an electron which has been received by the which has been donated by the reduced NAD. So and then they're going to be transferring them to the next component. So NAD dehydrogenase or NAD oxidoreductase is going to be an enzyme which is going to be helping in the oxidation of NAD to NAD. So this reduced NAD, so dehydrogenase, hydrogen is going to be removed. So this is an enzyme, or oxidoreductase. Why are we calling it NAD oxidoreductase? Because oxidation and reduction occurs simultaneously. So a complex consists of very many molecules which have been combined together. So we're having an enzyme, we're having proteins, sometimes some textbooks call them flavoproteins, and then iron sulfur containing proteins. So they are proteins which contain iron and sulfur. So complex number 2, that is succinate dehydrogenase. So succinate dehydrogenase is an enzyme that converts succinate to fumarate. If you look at the Krebs cycle, go back to the Krebs cycle and see the conversion of succinate to fumarate. So it is succinate dehydrogenase. So complex number 2. We are going to see the use of complex number 2 as we go to the electron transport chain. So complex number 3, we're having proteins containing cytochrome B and then cytochrome C1. So complex number 4, we're having cytochrome C oxidase, and then we're having protein complexes, that is A and then A3. So protein complex 4 contains an enzyme, cytochrome C oxidase, and then it also contains proteins cytochrome A and then cytochrome A3. So these are all electron carriers, complex 1, complex 2, complex 3, and complex 4. Now, number 2, we are having what we call mobile carriers. What is a mobile carrier? Mobile carrier is capable of moving from one point to another. So, previously we looked at electron carriers. These electron carriers are not the same as mobile carriers. These electron carriers may just receive and then later on give to a mobile carrier. The mobile carrier is capable of moving or crossing the phospholipid bilayer and then from one electron carrier to another. So these electron carrier complexes, they may be fixed in one position, but then the mobile carriers, they are going to move with the electrons. So the mobile carriers will have, number 1, we're having what we call ubiquinone. Ubiquinone, sometimes you can call it coenzyme 2. In animals, coenzyme 2, we find the coenzyme 210. So coenzyme, so there is coenzyme 2. So coenzyme 2, sometimes you can call it ubiquinone. So the reduced version of ubiquinone, we can call it ubiquino. So that is the reduced. So reduced version, that is the ubiquino, which is carrying an electron. So if ubiquinone receives an electron, it is going to be reduced to form what we call ubiquino. So it is the reduced version of ubiquinone. So another mobile carrier we have is cytochrome C. Cytochrome C, sometimes you can abbreviate it as site C. Site C. So it is also a mobile carrier. These two are not proteins. These two will not be proteins. They are just mobile carriers. They are not protein complexes. So they are not protein complexes, as you can see. The electron carrier complexes: complex 1, complex 2, complex 3, and complex 4. So let us look at the real process that is taking place. So this is our mitochondria. Our mitochondria, as we discussed earlier, we said that it has the outer membrane, it has the inner membrane. All the components of these membranes are the same because they all consist of a phospholipid bilayer. So the phospholipid bilayer is going to be found in the outer membrane, is also going to be found in the inner membrane. So now this space is going to be called the intermembrane space, and then inside here we are going to be finding the mitochondrial matrix where the Krebs cycle occurs. So outside glycolysis, that is the cytoplasm. So outside the mitochondria, we are finding the cytoplasm. So let us imagine a cell has been drawn surrounding the mitochondria. So outside, that is the cytoplasm where glycolysis is going to be occurring. Then the inside, that is the matrix. We are not going to be calling this the cytoplasm. We are calling it the mitochondrial matrix. That is the Krebs cycle. So inside the inner mitochondrial membrane. So this is the inner mitochondrial membrane. So we have two membranes, the outer and then the inner. So the inner mitochondrial membrane, that is where the real electron transport system, that is where the electron transport system takes place, and that marks the end. That marks the end of respiration. So let us look at from the inner mitochondrial membrane. Let us zoom it and then form this phospholipid bilayer. So I've just obtained only a small component of the inner mitochondrial membrane. Then I have zoomed it to form this phospholipid bilayer. So of course we know very well how a phospholipid bilayer looks like. So it consists of very many proteins, carrier proteins. It consists of the hydrophobic tails. It is very many components, as you can find out what a phospholipid bilayer is. So, but of course, I'm going to describe using the phospholipid bilayer of the membrane. So, in our membrane of the mitochondria, in our inner membrane, we're having complexes. So this is complex number 1, as we had earlier on mentioned, complex number 2, then complex number 3, complex number 4, and then complex 4 complexes have been here. So we are going to also look at the other one. So so the complexes that have been mentioned, you can see that complex number 1 contains FMN Flavin Mononucleotide. Flavin Mononucleotide is just a protein which is going to help in carrying the electron from one component to another. So FES, it is also another protein, a carrier protein. Of course, we say that a protein complex consists of very many proteins combined together. So this is the first complex. So the second complex is complex number 2. We are going to see that complex number 2. We say that it contains succinate dehydrogenase. So complex number 3, what do we have in complex number 3? We're having cytochrome B and then cytochrome C. So B and then C, that is complex number 3. So a combination of proteins forms a complex, a protein complex. So protein complex number 3. So protein complex number 4, protein complex number 4 is going to be containing cytochrome C oxidase, cytochrome C oxidase. So cytochrome C oxidase, we're going to see its role, and then cytochromes A and then cytochromes A3. Cytochrome A and A3, these are just pure proteins, of which these cytochromes A and A3, when you combine them, you form a protein complex 4. But of course, we're having an enzyme, cytochrome C oxidase. Of course, we know enzymes are protein in nature, so still we're going to be having an enzyme there. So after finishing the complexes, we can look at the mobile carriers. So the mobile carriers, we're having cytochrome, we're having ubiquinone. So the first mobile carrier, we're having ubiquinone, that is acetyl, that is coenzyme 2, that is coenzyme 2. So this is ubiquinone, coenzyme 2. When you see CO2, this is ubiquinone. So ubiquinone is not going to be part of the complexes. So ubiquinone it is just a coenzyme. So, and it is a mobile electron carrier. So it is going to be carrying an electron from the first complex to the third complex. So it is also going to be carrying an electron from the second complex again, because this complex is going to release an electron and then take it to the third complex. So from ubiquinone, of course, we know that the reduced form of ubiquinone is ubiquinol. So we can look at cytochrome C. Cytochrome C, it is another mobile carrier. So this cytochrome C is going to be receiving an electron from the complex number 3 to complex number 4. So of course, we have now learned, we have seen that we're having 4 complexes and we're having 2 mobile carriers. So the mobile carriers, that is ubiquinone, that is CO2, and then site C. This abbreviation is representing cytochrome C. Those are all mobile carriers. Complexes, we're having complex number 1, complex number 2, complex number 3, and then complex number 4. We have seen what we find in those 4 complexes. So lastly, we look at the ATP synthase. ATP synthase is still another protein, but now this ATP synthase is going to be acting as a pathway for the protons from outside or from the intermembrane space. Now, if you look at this phospholipid bilayer, this space here, we can call it the intermembrane space. So this is the space here. So this intermembrane space. So this is the intermembrane space. So it is the intermembrane space. So just imagine there is an outer membrane, and then this is the inner membrane, which is the phospholipid bilayer. So this is the intermembrane space. Whichever components you are seeing outside here beyond this, that is those components are in the intermembrane space. So let us start breaking down the electron transport system. Now, if you look at our phospholipid bilayer, contains very many components. But now, out of those components, we're having protein complex number 1. This protein complex number 1, it is going to contain NADH dehydrogenase or NAD oxidoreductase. NAD oxidoreductase, it is going to oxidize NAD. NAD at this stage is reduced. We know very well reduction, it is the addition of hydrogen. So this NAD has hydrogen and it is reduced, or it is the addition of electrons. So it means that if NAD is reduced, it means electrons, it is electron rich. So it means that if it is electron rich, it is going to donate electrons to protein complex number 1. So if it donates electrons to protein complex number 1, of course it is going to become oxidized. Oxidation is the loss of electrons or removal of hydrogen. So hydrogen has removed, electrons have been lost. So these electrons which have been lost are going to be picked up by the protein complex number 1. So this protein complex number 1, these electrons are going to start traveling through a series of proteins which are found in protein complex number 1. So the first protein to receive that those electrons, that is Flavin Mononucleotide, FMN. Flavin Mononucleotide then transfers those electrons that have been received to iron sulfur containing proteins. So within the complex number 1. Remember, NAD has been reduced and then hydrogen ions have been removed. So now, these hydrogen ions which have been removed, they are going to be pumped. They are going to be pumped to the inner to the intermembrane space, and this intermembrane space is going to be charged. So 4 of them are going to be taken to the intermembrane space. So the pathway that is going to allow these hydrogens to cross, it is only going to be this protein complex. So it is very impossible for these hydrogen ions to cross via the via these hydrophilic tails, because they are not polar. So the only pathway that we are going to have for the crossing of the hydrogen ions is going to be the protein complexes. So when you see this arrow, just show that the hydrogen ions or the proteins have crossed via a protein complex number 1. So when you look at these red arrows, they show how our electron is moving. So this electron, after moving from the iron sulfur containing proteins, it is going to be sent to ubiquinone. Ubiquinone, sometimes you can call it coenzyme 2. So the iron sulfur containing proteins donates it to ubiquinone. Ubiquinone, after receiving that electron, it is going to be reduced. So once it is reduced, it forms what you call ubiquinone. Ubiquinone is a reduced version of ubiquinone. So this ubiquinone is also going to receive electrons from complex number 2. Complex number 2 is going to receive FAD, which is reduced FAD, and then this FAD is going to be oxidized to form oxidized FAD. So this FAD joins later the ETS. So it means that if it joins later, it implies that there is less energy which is going to be released. Remember, the more the electron moves, the more energy that is going to be released. So this energy this electron that is released here by FAD, it is released at a lower energy level than the ones which are reduced, than the one which is released by NAD. So NAD produces more energy than FAD. So if NAD produces like 3 ATP molecules, then FAD is going to produce 2 ATP molecules. Why? It joins the electron transport system later on by donating electrons to complex 2. So after donating these electrons to complex number 2, these electrons are going to be transferred to ubiquinone, and then ubiquinone is going to become more reduced. Again, we are going to remain with ubiquinone. So ubiquinone is going to be highly electronegative. So these molecules, each molecule that is receiving an electron at a higher energy level is more electronegative than the other. So iron sulfur containing proteins are more electronegative than the Flavin Mononucleotide, and then ubiquinone is more electronegative than the iron sulfur containing proteins. So ubiquinone, now we are having ubiquinone which is electron rich, transfers. Remember, it is mobile. it is capable of crossing the phospholipid bilayer from complex number 1 to complex number 3. So complex number 2, there is no way it is going to pump hydrogens to the intermembrane space. So for it does not pump electrons in the intermembrane space. So it is only complex number 1, complex number 3, and then complex number 4 that pump electrons to the intermembrane space, creating a proton gradient or an electrochemical gradient. So coenzyme 2 then transfers electrons to complex number 3. Complex number 3 consists of cytochrome B and then cytochrome C. Cytochrome C is more electronegative than cytochrome B. So cytochrome B is going to receive the electron first, then the electron is going to be shifted to cytochrome B within complex number 2. Of course, we are going to have hydrogens, 2 of them shifted to the intermembrane space. So the intermembrane space, still, these hydrogens are capable of crossing up to the cytoplasm where glycolysis is occurring. So it means that the outside of the inner membrane is more charged than inside the matrix. So this is the inner membrane. So this is the intermembrane space. So more charge and then less charge. That one alone creates an electrochemical gradient. That electrochemical gradient is going to create a potential difference. So if we have 4 hydrogen ions out here, 2 of them, and then 4 of them, in total creating 8, that one alone creates an electrochemical difference, or a potential difference, because we're having more positive here and then less positive or negative this side. That one alone creates a potential difference, of which we are going to have electrical energy generated. Of course, again, as we are seeing the movement of electrons from proton complex number 1, number 3, number 4, that one alone, the movement of electrons, we know very well by definition what is current. Current is the flow of electrons. When electrons flow, also current flows, and current produces electricity. So this energy which is produced within complex number 1, complex number 3, complex number 4 is used to pump hydrogen ions into the intermembrane space. So energy is released in complex number 1, complex number 3, and then complex number 4. That energy is used to pump the protons in the intermembrane space. But now, when they are pumped in the intermembrane space again, they are going to create a potential difference between the outer membrane space, between the intermembrane space, and inside the matrix. So inside the matrix, that is the site for the Krebs cycle. So from cytochrome C, we are going to have this is cytochrome C1. Can make that correction. Cytochrome C1. So cytochrome C1, that is complex number 3. So complex number 4 consists of cytochrome C oxidase, and then that is an enzyme, and then cytochrome A, and then cytochrome 3. So from complex number 3, we are going to have our electrons picked up by cytochrome C. Cytochrome C is a mobile carrier. So it is capable of crossing the phospholipid bilayer, unlike the complex cannot cross. So it can just donate the electrons to cytochrome C. So cytochrome C, after receiving the electrons, it becomes reduced. So it is going to move across the phospholipid bilayer to complex number 4. So complex number 4, it contains cytochrome C oxidase. Now cytochrome C oxidase is an enzyme that oxidizes cytochrome C, which is the mobile carrier, by removing electrons from it. So that these electrons are taken to complex number 4. So it contains cytochrome C oxidase, while it oxidizes cytochrome C, which is the mobile carrier. So moving further, when you go to complex number 4, complex number 4 has cytochrome A and then cytochrome A3. So cytochrome A and cytochrome A3, A3 is more electronegative than A. So A receives the electron first, and then this electron is donated to cytochrome A3. So at that stage, that is the end of our movement of the electron, and at this stage these electrons are going to be received by oxygen, which is the most electronegative at that stage, and that is showing how significant is aerobic respiration, because this oxygen is coming from the air that we breathe in. So if you don't breathe in, it implies that you will not get the oxygen that will complete the electron transport chain. Remember, the electron transport chain produces the biggest percentage of the ATP that we use in our body. So if you block this oxygen from entering into the body, then it means that little ATP is going to be reduced, is going to be produced from the Krebs cycle and then the glycolysis. And from glycolysis. So it implies that the final electron acceptor in the ETS, or in the electron transport chain, that is oxygen. So electrons are received by oxygen and then two hydrogen ions, then to form water. But now, what is the source of the two hydrogen ions at this stage? So remember, we said that we're having proton pumps inside our complexes, complex number 1, number 3, and then number 4. So they use energy created by the movement of electrons from complex number 1, complex number 3, complex number 4. So if we are to look at the electron transport system, the source of the hydrogen ions came from the hydrogen ions which are in the intermembrane space. But these hydrogen ions could not cross the phospholipid bilayer because of the hydrophobic tails. So the only pathway that could be used to allow these hydrogens to cross into the mitochondrial matrix, because now this is the mitochondrial matrix, it is the ATP synthase. At that stage, this ATP synthase, after hydrogen ions crossing, of course, an electrochemical gradient is going to exist and energy is going to be released. So at that point, when energy is released, we won't have ADP combining with an inorganic phosphate found within the mitochondrial matrix to form ATP. So the process is called oxidative phosphorylation because oxygen is involved, and phosphorylation takes place in presence of an inorganic phosphate to form ATP. So at that stage, we are seeing more energy formed in the electron transport chain. So any NADH would form 3 ATP molecules, but now the NADH which comes from the from oxidative decarboxylation in the link reaction and glycolysis, plus the one which has been formed. So the ATP molecules which have been formed from the link reaction and glycolysis, for them to cross this from the cytoplasm through the outer membrane into the intermembrane space, they use energy. So one ATP molecule is going to be used for the NAD to cross from the outer membrane into the intermembrane space so that it can be used in the electron transport chain. So it means that one ATP is going to be consumed. So it implies that if each NAD produces 3 ATP molecules, we expect each NAD to be producing 3 ATP molecules in the electron transport chain. But since one is used, minus 1 ATP, since one is used, so 1 ATP is used, so we shall subtract that 1 ATP which is used for the NAD to cross from glycolysis into the membrane of the mitochondria into the inner mitochondrial membrane. But now the NAD that is coming from the Krebs cycle, it is going to give purely the 3 molecules of ATP. Why? Because there is no energy that is required for this NAD to come and then be reduced, be oxidized to form oxidized NAD. So remember this matrix, that is where Krebs cycle occurs. So NAD is going to be readily available here. So it will just come and then get oxidized from there. But now the NAD which is coming from the cytoplasm, for it to cross the membrane, it is going to require a lot of energy. And the 3 ATP molecules, which could have been formed from the NAD which is coming from glycolysis, instead we are going to form 2. But the NAD which is coming from the Krebs cycle, we are going to form a net of 3 ATP molecules. But from the glycolysis and the link reaction, or from the other oxidative decarboxylation, that is the link reaction and glycolysis, we are going to form a net of 2 ATP molecules from each NAD that is crossing the membrane. So it implies that if we are to look at it, we are to calculate the NAD that is coming from the glycolysis and the link reaction. So each NAD is going to give a total of 2 molecules. But the NAD which is coming from the matrix, that is the Krebs cycle, is going to give a total of 3 molecules, giving us a total of 5 molecules from one NAD from the Krebs cycle, and from one NAD from the link reaction, and another one, if at all we are also having another one from glycolysis. So from glycolysis and link reaction, each NAD is going to give us 2 ATP molecules innate. Why? Because each NAD is expected to give us 3 molecules, but because 1 ATP is used for each NAD that is crossing the membrane, the membrane of the mitochondria, it implies that you are going to get a net of 2 molecules of ATP, like that. So we go to FAD. FAD produces less energy because it joins the electron transport system later on. So each FAD molecule is going to be producing 2 ATP molecules. But remember, FAD doesn't require any energy to cross because it is already found in the mitochondrial matrix. So it will instead just donate its electrons to complex number 2, and then those electrons are going to be carried later on to the next carriers. So a summary of electron transport system: NAD donates reduced NAD donates electrons to complex number 1. Of course, we have seen that. Step number 2, reduced FAD donates electrons to complex number 2. So electrons move through the chain releasing what? Energy. So once these electrons are moving through the chain, they will release a lot of energy. Of course, that energy, I've told you what is the use of that energy. It is going to be used to pump protons into the intermembrane space. So the intermembrane space is more charged than the inner matrix, so creating a potential difference. Once that potential difference is created, it is again going to create electrical energy. That electrical energy is going to allow these protons to again cross the ATP synthase and then back to the mitochondrial matrix, so that water, it can allow the formation of water via oxidative phosphorylation. So energy pumps protons into the intermembrane space. Protons flow back through ATP synthase, forming ATP. But as well, I'm going to put the description of the full electron transport system in the description down there, so that in case you need a full description of what is happening here, or what I've talked, because not whatever I've talked is available here. So you can use that description again to further understand the electron transport system. But again, if you have any more questions, you can still write down there in the comments. I'll be able to answer you. So that is all about the electron transport chain. So let us calculate together the net analysis of the total ATP molecules that are formed, so we can make these calculations together. So if we are to make these calculations together, let us first look at the analysis from glycolysis. From glycolysis, if you look at our previous lectures, we realize that we have 2 ATP used initially. Yes, you can go back to the previous videos, you can check, and then you see 2 ATP molecules used initially, and then 4 ATP molecules produced. So it means that if we are to get the net ATP molecules that are produced, we are going to get 4 minus 2, giving us a total of 2 ATP molecules. So when 2 ATP molecules are produced, again we have 2 NAD molecules, 2 reduced NAD molecules being formed. I have told you that these reduced NAD molecules, we are going to later on see them in the electron transport chain, helping us to generate ATP. So in the link reaction, I remember discussing it, that in the link reaction, that is why we're having oxidative decarboxylation of the pyruvate. So that pyruvate, the molecule is reduced by removing a carbon and then some molecules of oxygen, so forming carbon dioxide somewhere. So oxidative decarboxylation of the pyruvate takes place at that stage. And what takes place is that we have only 2 reduced NAD molecules being released. Remember, carbon dioxide is also produced, but is not important at this stage because it is released as you are breathing out. So the most important molecule is the reduced NAD. So we're having 2 of them. Why are we having 2 of them? Because 2 pyruvate molecules are decarboxylated. Remember, from glycolysis, we end up with 2 pyruvate molecules. So these 2 pyruvate molecules, each pyruvate molecule is going to produce a single NAD, because each pyruvate molecule is going to undergo a single oxidative decarboxylation. So the 2 molecules are going to produce 2 reduced NAD molecules at that stage. So entering into the Krebs cycle, that is the second stage, we're having 2 ATP molecules. Of course, we know the ATP in some in plants we find there GTP, guanine triphosphate. And now let us just call it ATP for the sake of animals. And then we're having 6 NAD molecules produced. Remember, I told you each cycle produces 3 molecules of NAD, of reduced NAD. But since there are 2 pyruvate molecules which are going to be decarboxylated to, and later on coenzyme A is added to form acetyl coenzyme A. So we have 2 acetyl coenzyme A molecules being taken into the Krebs cycle. So each molecule is going to produce 3 reduced NAD molecules. So if we are to double them, then we are going to form a total of 6 reduced NAD molecules. So if we are to go to FAD, reduced FAD, each cycle of the Krebs cycle produces 1 reduced FAD molecule. But since there are 2 acetyl coenzyme which are entering into the cycle, then we are doubling our FAD into 2. I remember giving you that analysis in the Krebs cycle, so we can go back and check it in case we have forgotten. So our main intention is to look at the analysis of the ETC, that is oxidative phosphorylation. So in other words, we are going to look at how many ATPs in total shall we form. So remember, in the previous lecture, we realized that there is pumping of protons across the membrane using so these are hydrogen ions. All these are protons of hydrogen using protein complexes 1, 3, and 4. So these protons, the more that are pumped, the more energy we expect to produce. So remember, these protons here, NAD pumps more protons than FAD, because FAD enters later in the chain. So we need to look at the analysis of what NAD produces and what FAD produces. But before that, we can first look at the total NAD that enters into the chain. So we can first begin by counting. So NAD. So how many NAD do enter the chain? So if you look at it, how many do we have in the glycolysis? We're having 2 of them. So we're having 2 from glycolysis. So from glycolysis, we go to the link reaction. To the link reaction, we have 2 still. Then from the link reaction to the Krebs cycle, we're having 6. So they are we have 2 plus 6 reduced NAD entering into the electron transport chain. So giving us a total of 10 reduced NAD. So having a total of 10 reduced NAD. So We can go to reduced fat. We count them. How many do we have reduced fat? So in glycolysis we don't have any. In the link reaction we don't have any. And in the Krebs cycle we have a total of 2 reduced fat. So we only have 2 of them. So now from this we can form a basis of getting how many ATPs are obtained in the electron transport chain. I told you that the electron transport chain produces the biggest percentage of the ATP molecules. So what determines the ATP molecules that are produced? The protons which are pumped into the intermembrane space. So NAD pumps more proteins than FAD. So NAD pumps 10 hydrogen ions. 10 hydrogen ions. But remember, the hydrogen ions at a certain stage, they cross the ATP synthase. They cross the membrane via the ATP synthase, and 4 hydrogen ions that cross, they form a single ATP molecule. Each 4 hydrogen ions that cross form a single ATP molecule. So it implies that if a single NAD pumps 10 hydrogen ions, then and 4 hydrogen ions form a single ATP molecule. So it means that each NAD, it means that 10, because we're having 10 NAD molecules are going to give us how many hydrogen ions? We are going to have 10 times 10 hydrogen ions, so giving us 100 hydrogen ions. So if 10 NAD molecules give us 100 hydrogen ions, so it implies that we can easily get how many ATPs can be obtained from the 10 NAD molecules, or as a result of pumping 10 hydrogen ions. So you can just get 100 ions divided by 4 ions, you can get the total. So giving us 100 ions divided by 4 ions, so giving us a total of 25 ATP. So giving us a total of 25 ATP. So 25 ATP are formed from 10 NAD molecules. So 10 NAD molecules form 25 ATP molecules. So how about the FAD molecules? The FAD molecules, FAD, reduced FAD pumps 6 hydrogen ions. Each reduced FAD pumps 6 hydrogen ions. And remember, I've told you that each 4 hydrogen ions that cross the ATP synthase, they lead to the formation of a single ATP molecule. So it implies that we are going to just get 6 hydrogen ions divided by 6 hydrogen ions divided by 4 hydrogen ions. So in total, we are going to have a total of 1.5 ATP molecules. 1.5 ATP molecules are going to be formed as a result of the FAD entering into the electron transport chain. So it implies that if you are to add the total ATP formed in the electron transport chain, we are going to have 25 plus 1.5, which is going to give us 26.5. If 26.5 are formed, then later on we can see later on we can add the total. So if we are to add the total ATP formed, we are going to begin from glycolysis, Krebs cycle, and then later on to the electron transport chain. So we can start adding at that stage. So if we have a single FAD molecule producing 1.5 ATP, then what about 2 FAD? Because if you look at the Krebs cycle, we're having 2 of the reduced FAD. So it implies that if you're having 2 reduced FAD at that stage, we're going to have 2 times 1.5, so giving us 3 ATP. So giving us 3 ATP. So 2 FAD from Krebs cycle will give us 3 ATP in the electron transport chain. So it implies that we shall add 3 plus 25, giving us 28. So the ETS of the electron transport chain gives us a total of 25 plus 3, giving us 28 ATP molecules. 28 ATP molecules. So overall, so you can get the overall. Overall, that is the ATP from glycolysis, stage 1, stage 2, that is the link reaction, stage 3, that is the Krebs cycle, then stage 4. So I'm describing this as stage 1, then stage 2, stage 3, and then this is stage 4. So there are 4 stages, though there are mainly 3 stages, but now the other stage which comes in and fixes in is the link reaction that links the glycolysis to the Krebs cycle. So we can count the overall ATP molecules counted from stage by stage. So the first stage is the glycolysis. Glycolysis produces a net of 2 ATP. So 2 of them. So we have seen them here. Then in the link reaction, we have 0. We have no ATP produced there. Then in the Krebs cycle, we're having a total of 2. Then lastly, in the electron transport chain, we have seen NAD molecules which entered into the cycle, into the chain. There are 10 of them, giving us a total of 25 ATP molecules. And then to FAD, a single FAD produces 6 hydrogen ions, and then the 6 hydrogen ions divided by 5 hydrogen ions gives us a total of 1.5 ATP. But remember, 2 FAD molecules were used, so it implies that we're going to multiply 2 times 1.5, giving us 3 ATP. So overall for the ETS we got 28. So 28. So the ATP. So it implies that we're going to add 2 plus 0 plus 2 plus 28. So someone can add and give us the answer. So if we have 28 plus 2, we are going to get 30, and then plus 2, we are going to get 32 ATP molecules. So overall ATP molecules which are produced are going to be 32 molecules of ATP. So what you can see is that the electron transport chain is very significant. It implies that it produces a lot of ATP. So what may block it from continuing? It is the absence of oxygen. So in any case, the electron transport chain can never be anaerobic. It is always aerobic. So oxygen is very crucial at that stage. So if oxygen is not available, because it is the final electron acceptor, then energy may not be released and the electron transport system will stop. But in the previous lecture, looked at glycolysis and the Krebs cycle. These two can occur with or without oxygen, because in the Krebs cycle and the glycolysis, we don't see at any stage where oxygen is entering in, but it may just be used indirectly. So though even in its absence and aerobic conditions, still glycolysis can still occur. So though the only challenge is that glycolysis and Krebs cycle, they yield less energy than in the electron transport chain. So the electron transport chain is the most significant part of the cellular respiration because of its production of ATP, large production of ATP. So if you look at the calculations, it gives us more ATP than the rest of the other stages. So this is the inner mitochondrial membrane, and this is the intermembrane space where having hydrogen ions positively charged. So most of them are going to be pumped into the intermembrane space, and the pumping of them into the intermembrane space through the complexes, complex 1, 3, and 4. So this pumping, these hydrogens are always generated by these molecules, these NAD and FAD molecules. So once the single NAD alone will pump about 10 hydrogen ions into the intermembrane space. So at a certain time, we are going to have those protons again crossing the membrane via the ATP synthase, as we have seen in the previous lecture of the electron transport chain, and then they come back into the mitochondrial matrix. As they come back into the mitochondrial matrix, we are going to have an electrochemical gradient created, or a proton gradient created, which is going to produce energy. So that energy, which is the ATP, which is going to be formed at the same time. So each four molecules, each four ions of hydrogen that are going to cross the ATP synthase via the ATP synthase that are going to cross the membrane via the ATP synthase are going to produce one ATP molecule. So if one reduced NAD molecule pumps 10 ions at a time, what does it imply? It means that each NAD molecule is going to produce how many ATPs? We're going to have 10 divided by 4 ATP molecules, giving us about 2.5 ATP molecules. Though I've just given a full calculation, a simple calculation here to see how many hydrogen ions are going to be pumped. So if you have 10 NAD molecules, 10 reduced NAD molecules, they are going to pump 10 times 10 hydrogen ions, giving us 100 ions. So the 100 ions in total can give us 100 divided by 4, giving us a total of 25 ATP molecules. Lastly, we can have FAD. A single FAD alone produces less hydrogen ions, of which it produces 6 hydrogen ions only. So the 6 hydrogen ions, when you divide them by 4, it will give you a total of 1.5 ATP. But remember, in the Krebs cycle, we had 2 FAD molecules being produced. So the 2 FAD molecules that we have produced, it means that each FAD molecule is going to produce a total of 1.5 ATP molecules. So 1.5 times 2, it gives us a total of 3 ATP molecules. So it means that we can get the overall ATP molecules which have been formed by getting 25 ATP formed by NAD, and then 3 ATP molecules formed by FAD. So if we add the 25 plus 3, then we shall get a total of what? 28. So the overall, we realize that 2 ATP molecules, the net obtained from glycolysis, Krebs cycle, we obtained 2. So if we have 2 plus 28, it gives us a total of 32. Thank you very much. In case you have a question, you can put in the comments.